And here you'll find all of the workflows that are part of this workspace. And the step I'm going to be showing you how I created is step 2, BAM sorting and filtering. So to begin with, I needed to understand what the goal of this step was. So my goals with creating this workflow was to use the output from the first step, which was star alignment, as input for this step. And I wanted to sort the BAM file that was used as input. I wanted to filter the BAM file and keep only passing WASP tags. And I wanted to index the new BAM file. All of this was to be done using SAM tools. So this is my the overview of what I wanted to accomplish using this step. So I'm navigating back to the Workflows tab, and I'm going to click on Find a Workflow. And when you do this, you'll see that there are some suggested workflows. There's a doc store, and there's also the Broad Methods repository. So I will click on Broad Methods repository. So to find a suitable starting point, I knew that I needed to use SAM tools. So in the search bar, I am looking for SAM tools. And this will give me access to all of the public methods that use SAM tools. And I'm going to try to find the best example that I can use as a template for what I wanted to accomplish. So by looking through what's available, I found the AMP-PD workflow, RNA, BAM, sort, and index. When I read through the documentation, I can see that what this workflow does is it runs SAMTools sort and SAMTools index on a BAM file. It then runs SAMTools flag stats on the old and new file and verifies that they're the same. So this does a few of the things I want to do. It runs SAMTools sort and SAMTools index. I don't need to run SAMTools flag stat, but I do need to add an additional filtering step. So this is a good starting point for modifying it to for my workspace. But if this is what you need to do, you can export it as it is to your workspace as well by pressing Export to Workspace right here. But since I need to make changes, I'm going to first clone the workspace, the WDL script. So the first thing I need to do is give it a namespace and a name. A namespace is a folder that contains different WDL scripts. So because I'm going to use this in the variant aware alignment workspace, I'm just going to name it BAA step 2 maybe. And then I can give it a name that is suitable for what I want the workflow to do. So because we're doing SAM tools, BAM, sorting, indexing, and filtering, I'll name it RNA BAM sort index filtering. So within the WDL script, there are a few sections. First, there's some documentation. And then we have our workflow variables that are defined. Variables are also defined at the task level. So that is these variables. The variables at the task level and the workflow level are connected to one another in this section. And the files that are created as outputs are defined here. All of the commands that are run within the script are found in the command section. And if we take a look at this particular script, there are a lot of echo commands that make the script a little more verbose. This is useful if when you take a look at your job manager to kind of understand where your script is at and what is happening. And then it looks like they're creating a soft link between an old BAM and the new BAM. They're running SAMTools sort, SAMTools index, and then running the steps for comparing the old and new files. There are also some runtime, the runtime variables are configured and the names of the output files are configured below. So to begin with, I think I'll remove the sections of the script that I know I will not be using, which would be, I don't want to create a symlink to the old BAM, so I can remove that section. I also don't want to do the comparison step, so I can remove the bottom section as well. So now I have two echo commands for samtools sort and samtools index. What I want to do is after sorting, I want to filter the BAM file that's been sorted. So I have that command written over here. I'll grab that. So first we're running samtools sort, and the output of this command is stored in this location. I want to use this file as input for my next step. So here I have two samtools view commands that are running, and the file that is being viewed is this BAM file. And I'm piping that into a grep function that is helping me keep only reads that have passing tags. And I'm finally storing all of that in a new location that is called samplename.waspfilter.bam. In the next step, I want to index this BAM file. So right now, the index step is actually, it's samtools index for the older BAM file, but I want to change that into the WASP filtered BAM file. So now we have samtools sort running on a file, and it's being stored in this location. Using that sorted file, we're filtering for passing WASP tags, and we're storing it in a new location called samplename.waspfilter.bam. And then I'm using that as input for the indexing step. In order to continue the pattern of having the script be verbose, I'll also add another echo command before we run the filtering step. Now, with that, we have a set of commands that accomplishes what I want it to do. Now we just need to configure our input and output variables to reflect the changes that I've made. So to begin with, our input is going to stay the same because we have a sample name and input BAM file, and those are the inputs I'm going to be using. Our runtime configurations don't need to change, but our output files are going to look a little different because I'm no longer creating these two output files. So I can go ahead and remove that. And I can do the same in the bottom section as well. So in this section where the output file paths are defined, this needs to reflect the new file name that we're using. So since this is my file name, I can change the name of the output to samplename.waspfilter.bam, and I'll do the same for the index as well. So I'll change the documentation in the top as well. And this documentation can be used to update documentation below by pressing populate from WDL. You can see that now we have our new files here. And with this, I've modified the existing Whittle script to to reflect what I needed to accomplish. And I can press create new method. And so now I'm the owner of the new method I've created, and I can export this workspace to uh, my copy of the workspace we're using. Our root entity type for this workspace is sample, so I can change that, and I can press export to workspace. So now in the Workflows tab, we have our new workspace present. If I click on the workspace, I can set up the different input parameters and so I can set up the different input parameters required for this run. I won't go into too much detail about exactly what we're using, but I'll put the input variables in right now. So the syntax of this.bam file lets the script know that it needs to look in the data table section under the column BAM file, and it will retrieve whatever is there and use that as input. For number of CPU cores, I think it's 2. And we're using 3 preemptible tries. Under runtime zones, I'm going to be giving it this option. And again, for sample name, I want to call whatever the sample name is in the data table. So I can say this dot sample ID. I will give it the link to the URI of the Docker. And I'll also enter the amount of memory and disk space required. By pressing save, this will save these changes. Under output, this is where you can define where in the data table you want your output files to go to. So for example, if I were to say this dot, this is for the index file, so I can say this dot BAI output path, and this will create a new column in the data table and put whatever output file is created into that location. I'll press save again. So now we have our modified script with the new command. We also have our inputs configured and we have our outputs configured. So we can select data and click on the samples that we want to process. So if I wanted to process these 2 samples, I can click on that and press OK and press run analysis. So if you wanted to process multiple samples at once, you can click on them and press run. And launch. So now it navigates to the job history tab where you can see the status of jobs. So right now it's submitted, and then while the job is running, it will say running, and when it's completed, it will say completed. And while the job is running, you can also check on it using the links provided here. So yes, with that completes my demo portion of this webinar. I'd like to end with some acknowledgements. I'd like to thank Dr. David Craig, whose guidance helped me complete this project, and I would like to thank Ivo Violich, who um, helped me troubleshoot along the way and who's been very supportive of me throughout this whole process. I would like to thank the folks at AMPD, specifically Willie, Barry, and Dave. When I faced different issues along um, when I was learning Terra, they provided some resources that really helped me move forward. Victoria has been a huge help in getting this webinar set up and just uh, everything relating to that. I'd like to thank my lab members at USC, Michelle, Mackenzie, Kayla, and Nina. They've heard this presentation a few times, and I really value their feedback. And finally, I'd like to acknowledge the NIH and their funding for this project. Thank you.
That was really great. Thank you so much for walking us through that and just how, I guess the steps it takes to set up a workflow. So that was really great to see kind of how you set it up, especially because I've seen the end products and I've run the end products, but I've never actually seen how to do it. So that was really great. Thanks so much for walking us through that. Were there any questions that anybody had? We were answering questions in the chat during the presentation. So if there's any additional questions, there were some questions regarding the cost of using Terra. So there were examples shown for the specific workspace that was being run and the cost of that. So thanks for sharing that, just so people get an idea of how much it costs to do some of these things. And then obviously the cost does depend on the workflows that you're using, whether you're running a workflow like this, or if you're running a notebook. Running notebooks typically less, I guess, costly. In general, running just the notebooks is slightly less costly than running the workflows. Let me see. And then just to clarify, these FASTQ files are AMPD FASTQ files. They are available through previous data releases. So if you go to the AMPD website, if you don't have Tier 2 access, you're able to register for Tier 2 access, and you're able to get access to the transcriptomics, the genomics, and the proteomics data with Tier 2 access. Were there any other questions that individuals had?
I had a question. This is Ivo.
Yes.
Ramya, you showed how you can sort of take a workflow that is similar to what you are trying to do and edit it. I haven't really worked in Whittle. I've sort of seen it a lot, and it's very confusing to me. So did you write any Whittle workflows from scratch? Did you use any other approaches, and how much time did you spend sort of working in Whittle and learning the syntax of it?
So initially for the workflows that I created, I did use pre-existing examples, but after I had used Riddled for a little bit, I understood the syntax a little better. And for example, for the Deep Tools script that I wrote, I didn't find an example that I could use as a base, so I did develop some scripts from scratch as well. I think there is a little bit of an initial learning curve, but once you understand the syntax, it's not too bad.
Were there any other questions? Okay, trying to figure out how to share this. So just a reminder of what was presented. So, getting into Terra and then finding the workspaces section, finding the specific workspace, and then cloning the workspace. She also went through all the parts of the workspace and then generating and running a workflow. And then you did also go into the job history tab, which I didn't add here, and talked about that a little bit. Were there any questions on any specific parts of the workflow or any of the data or anything like that? Thanks, Dave, for putting that link in there. Dave just put a link in the chat if anybody's interested in learning more about Whittle and how to learn how to use Whittle. Terra does have some great support resources in their, I guess, support section. So he linked that down below if you're interested in running those. Okay, and with that, I guess we'll move on to part 3. Were there any questions in general about, I guess, either the webinars or anything like that before we move on? And then once again, your feedback is pretty crucial. It'll help us understand, how to tailor these webinars as we continue expanding them and making them more public. So, for those of you who didn't have tier 2 access, do you think this would have been more beneficial if you did have tier 2 access? For those of you that did have tier 2 access, it'd be great to hear your feedback as well. So like I said, just getting some feedback from the survey, I put in the link, but it should take about 5 minutes to get through, and it would be really helpful, if we understand like what your current role is, whether you're a student, a postdoc, a PI, and your experience with data analysis. This will kind of help us gauge, how to make these webinars, as useful as possible, because our goal is to, generate all of these resources for people to use the data, but we also want these resources to be easy to use and accessible. And so, part of that is doing the webinar series, but we also want to make sure that we're tailoring the webinar series to make sure that everyone's able to get the most out of it. So this was today's webinar. We have some upcoming ones that should be pretty interesting. So the next webinar is May 11th, same, I guess, day and time. And it's going to be focusing on APOE status and looking at APOE status across AMP-PD subjects and cohorts. So we're going to be looking at APOE genotype for various subjects and cohorts within the AMP-PD data and combining clinical and omics data. For those of you that aren't as familiar with the AMP-PD data itself, it is made up of multiple cohorts that have been obtained from different projects. And they've been unified and essentially put up, for use. And so there are different cohorts. And so it's interesting to see, what is the APOE genotype for these different cohorts and different subjects within AMP-PD. So we have almost, Dave and Barry, correct me on this, but I think we have over 10,000 genome samples. And then close to that with transcriptomics and not as many proteomics, but.
So yeah, we have 10,400 subjects. We have whole genome sequencing for 10,400. We have transcriptomics data for about 3,500 subjects, but we have multiple time points for those RNA-seq samples.
Thanks. So these are the webinars that are coming up. If you are part of the AMP-PD community and you've generated any workspaces or notebooks or resources that might be of interest, feel free to reach out to me and we can get you scheduled to present on your resource within the webinar series. If this webinar has inspired you to use Terra and AMP-PD data and you want to present later on within the webinar series, also feel free to reach out to me and we can get you scheduled for that as well. And this is our thank you slide. It really does take a village for this project. This is a very large project, multi-site, I guess, multiple companies, public and private partnership, and lots of working groups. So just thank you to everyone who has participated, and a special thank you to all of those in the AMPD community who are generating resources like this one for others to use. And with that, I think that concludes our session. If there are any questions, we do still have a little bit of time, so I can stay on and answer any questions that anybody might have. If they're, whether they're AMPD related, Terra related, or anything like that. If there's any questions, don't all shout at once. Barry, Dave, did you guys have anything to add that I might have missed?
No, I like that you shared that this is a platform for people who are working with the data to get some exposure, to build upon the work of others. I think you hit all the key points, but to make it what you want it to be, that is our goal. So that survey feedback is super important. This is a pilot. We're gonna drive towards more public-facing webinars in the future. So you can help drive this in a direction that works for you.
Great. Yep. Okay. Well,
Hi, Victoria. I actually have a
yes.
Sorry, yeah, I'm new to this. So I'm not totally sure what exactly the platform's for. Is that, like you said, is people share the data or share their kind of software, or exactly what is that? Or you can provide somebody provide some kind of software package you can put your data in and analyze it. Exactly what it is. I'm just a little bit not sure what exactly.
So are you asking specifically about the Terra resources? Is that what you're asking about, or just the AMPD community in general?
In general.
In general. Okay, Barry, would you like to take this and talk about, I guess, the overall
Sure.
the community
Sure.
and focus on
would you mind flipping to the last backup slide? It's the last one on the deck. That might frame it a bit.
Oh, I see this one. This?
Yeah.
Yeah.
Mm-hmm.
Okay.
So AMPD has done an amazing job pulling together all of the omics resources with the clinical data and combining research data from 8 different cohorts. But when we did this, we also threaded the same participant identifiers and naming conventions through everything, so that you can combine the data very easily in analysis notebooks and workflows. But the research data sits at the bottom of the stack, and then Google Cloud Platform is what we're using to store it and access it through the Terra platform, which is what you saw today. On top of that, then AMPD has provided a set of getting started workspaces and the types of building blocks that are already working that you can pick up and clone and make your own and use them to put your own analyses together. In addition to the getting started materials, we've also created some sample notebooks and other examples that show how to actually work with the data together with clinical data or proteomics. Next session would be APOE. But what we really want is to get more of the what is it that the community is building, and that's what Ramia shared today. Was something that she built that was different than what we had planned as a program, right? So Ramia's resources are now available, and you can see what she's done with them, you can clone that workspace and modify it to suit your own purposes. And in this way, the stack doesn't end here. We have the workspaces, but you really layer community on top of that. And the work of others can build, or you can build upon the work of others. And that, I guess, in a nutshell, is the idea here. Did that help answer your question?
Somewhat. So basically, if I got it right, it means that you have a big pool of data from different cohorts. And there are a set of tools that can use to analyze them. And people can utilize both data there, and it's also the tool people already built, or you have built your own tool to carry out your own analysis. Is that more or less right?
That's exactly it. I mean, the key point in Terra is that these resources exist. They already work. So you don't have to figure out how to merge the data. Somebody's already done that for you. You can focus on the things that are most important to you, and that's generating the research.
All right. In that case, how do I know what has been done? Is there a kind of a catalog of what people have already done, so they don't have to duplicate other people's work, or build upon some other
Absolutely.
work?
Yes. So currently, you can go into Terra as soon as you get your Tier 2 access, and you can see a listing in Terra of all of the workspaces that are available to MPD researchers. And that list is fairly extensive by itself. One thing that we're improving probably in the next couple of months is to put an annotated catalog of all the notebooks and workflows that are part of these resources on the public site, so that you can see them, click into them, and then have that take you directly. But that catalog is coming soon. The resources are there today, but you would need to go into Terra and see the workspaces in a flat listing.
All right, thanks. That's very helpful.
Great.
And then to all of those, I did get a question in here about Tier 2 access. So if you already have Tier 2 access, I need to personally add you to a group to be able to access some of the workspaces, because we haven't publicly released them yet. So this one is not publicly released. And so I need to add you to a group so that you can see it. So if you have Tier 2 access and you have a Terra account, email me or just let me know what email you use for Terra, because most of the time it's not the email you're using for work, and we can get you added to these. And we will be publicly releasing the proteomics workspaces soon, I guess within the next few weeks or so. So those will be made available, and those will accompany the data that's already been released for the targeted proteomics. So.
Great, thank you.
Thank you. Okay. Were there any last minute comments or questions before we wrap up? Okay. Well, thanks everyone. Thank you to our guest presenter. And yeah, thanks to everyone who attended. And then once again, if you have the time, please fill out the survey just so we get a better idea of how we can adapt these to fit your needs. Awesome. Okay, well, thanks so much.
Yep.
Thank you.
Thanks,
Nice to meet you.
Thanks, Victoria. All right. Take care, everybody.
Bye.